Bismillah, Assalamualaikum warahmatullah. You're listening to microbiology lesson on microbial applications in industry. This episode is part 2 of the topic. We're going to look at specific examples of microbial applications in two major industries, the health industry and the food industry. When it comes to health industry, the first thing that comes to mind is the mass production of biomedical products. And we use microorganisms as biological factories for many of these products. The first example is antibiotics. Antibiotics are substances that can kill other microorganisms. Well, kill or at least inhibit the growth of other microorganisms. Examples are penicillin, gentamicin. The thing is, many microorganisms produce antibiotics. It's part of their defense. There was a study where scientists put streptomyces in two different environments. So one is rich in nutrients, the other one is poor in nutrients. They found that when the nutrients are readily available, the bacteria produce less antibiotics. And when the nutrients is scarce, the bacteria produce more antibiotics. So we think it is a competitive mechanism against rival bacteria group. So the point is bacteria produce antibiotics all the time, but that doesn't mean we can use all of them industrially. I read that only around 1% of antimicrobial substances that have been discovered are commercially viable, that you can industrialize their productions. So it's not that easy to industrialize these biological products. The same goes to hormones, vitamins, vaccines. For this subtopic, I want you guys to focus on industrial productions of vaccines. I will leave a link to a video, so study that and then be ready for any questions from it. Look at each step and make notes of the details, right? So for example, step 1 is the production of antigen-producing organisms. So write down what are for example, the parameters that they pay attention to, the pH, temperature, homogeneity. Step 2 is extraction and purification. So in the first step, we grow the bacteria cells or viruses that produce the antigens. In the second step, we take out the bacteria or the virus and then we sort of clean them up to make sure that we are left with just the bacteria that have the antigens that we need, and there is no other stuff that were present in the media. The third step is inactivation, or in other places they call it suppression. Here, we kill the bacteria or virus so that they can't cause diseases when we administer it to the patients. But we don't want to completely destroy them because we need them, or more specifically, we need the antigens on them to still be able to trigger our immune system. In step 4, we start mixing the extracted cells with, for example, adjuvants or preservatives or other antigenic substances. We do that, especially, we combine it with other antigenic substances, usually when we are assembling multivalent vaccines. Multivalent vaccines are those ones that have multiple antigenic types. The idea is, if we can combine the antigens, for example, 2 different diseases in one vaccine, we don't need to give 2 separate vaccine shots to patients. We just need 1. I think the highest valence so far is 6. So imagine how cool that is. Instead of getting 6 different vaccine shots, you just need 1, right? Good news for those who don't like needles. So that's step 4. For step 5, we have the filling and preparation phase. Depending on the vaccines, we need to prepare them in liquid form or powdered form. So for example, we can freeze dry the vaccine to remove fluid, which in some cases improves the vaccine stability so that they're going to have a longer shelf life. And then step 6, we do batch releases. So once you have prepared the vaccines, they're going to get inspected. And part of that inspection is we take random samples, random bottles from the assembly lines. And then we run experiments to check for its efficacy and safety as well, especially safety. And this is an important point. People who don't have the opportunity to study microbiology like you guys are more susceptible to fear. They are afraid that the vaccines are going to harm them more than the diseases themselves, right? But remember, these massive companies, they need to take crazy level of safety precautions. Even if you are cynical, even if you don't believe that they care about your safety, you've got to believe that they care about money. I mean, if their vaccines hurt people later, their stocks are going to crash. They're going to get fined by the governments for millions of pounds or face a class action lawsuits, by the patients. And their brands are going to go down the toilet. It's going to make it harder for them to even get a new contract in the future. So that prospect is horrific for those scientists and officers who are in charge of producing these vaccines, you see. They can easily lose their livelihood if that ever happens. So they're probably more scared and more careful than we are with the things that, we cook and put into our mouth every day. So yeah, it doesn't mean vaccines are perfectly safe. Nothing that humans do is perfect. But, most often it's safer than suffering the diseases. That's step 6. And finally, step 7, they will distribute the vaccine. At this level, they'll have to maintain what's called the cold chain transportation, right? So the cold chain transportation means that the vaccines have to be moved around at the temperature of between 2 to 8 degrees Celsius. Yeah, they have to do this like all the time, right? In the trucks or on the planes, on the ship across the planet, doesn't matter. They have to maintain that cold chain right until the vaccine gets to the client. Okay, that was an example of industrial production. Now, for all different bioproducts that involve microorganisms, the industrial players have to pay attention to optimization. And they have one extra challenge compared to industrialists of the non-organic products, like cars or laptop. Why? Because they work with living cells, which can get infected, which can get contaminated during their manufacturing processes. So most of the time, the production environment is aseptic, and they'll use computerized monitoring system to make sure that, there's nothing weird going on with the cells or the media. The monitoring system is typically part of the bioreactors where you do the fermentations of the biological products. There are at least 2 types of fermentation: batch fermentation and continuous fermentation. In batch fermentation, everything is added and fermented, right? Then when you're done, the product is retrieved. And then what we do is we sterilize the whole thing, we clean everything up, and then put a new batch in. In continuous fermentation, we don't do that, right? We keep the bioreactor running continuously. We keep adding fresh media, nutrients to feed the microorganisms in the reactor. And then we take out the products at the other end, also continuously. So industrial fermentation using bioreactors generally involves 4 steps. Well, in the more advanced courses later, you can learn the specific steps within these big 4 steps. and you will learn also the enzymes and the mathematics of running a successful bioreactor fermentations. But for now, just do some cursory readings about these 4 steps, okay? Number 1 is the introduction of the microorganisms. Number 2 is the fermentation itself. So you put the microbes, then you put the substrates that you want to convert into products. And then step 3 is the downstream processing, perhaps adding some enzymes to accelerate the process, or optimizing the parameters like temperature, O2 saturation and stuff. And then step 4 is the removal of waste so that it doesn't contaminate your bioreaction. Bioreactors come in different shapes and sizes. They can get quite big. They can go more than 10,000 liters in capacity. I think Samsung's got one of those. They do biologics as one of their, what's it called? Subsidiaries, right? So Samsung started just by selling groceries in Korea. Did you know that? Right? Now they are a massive conglomerate. Okay, so bioreactors. They have different shapes and types. So at this point, you just study a basic template of the components. When you master the basics, later on, when you need to study a specific type of bioreactor for your PhD or for your work, it will come easier for you. A bioreactor has a tank as the reaction container. And the tank is covered by a thermal jacket, which, as the name implies, the jacket is to keep the temperature stable. And inside the tank, you have the agitation system, which is for stirring your reaction content around. It helps with the distribution of gas and substrates. A bioreactor will have at least 2 openings. One is the inlet opening with a feeding pump. This is where you insert your medium, your microbial cultures, and other starting materials. The second one is the outlet opening, usually at the bottom. So this is where you take out your effluent, your products of the bioreaction. So those are the big ones, the big components. The other components are for essentially maintaining the active reaction inside the tank. So you have, for example, an air supply pipe, and it's connected to a submerged aerator at the bottom. So this oxygenates the content of the tank so that your microorganisms can breathe. And finally, you usually have a couple of minor but critical components like sensor probes. So these probes are connected to a system monitor outside, connected to a computer. We use them to sense the gas level, the temperature, the pH, and other parameters depending on the bioreactor model that your company purchased or your lab purchased, and also the types of reactions that you want to run in them. All right, so that's a bioreactor. Another industry where microbiology is applied is water treatment, especially wastewater treatment. I'm going to stop after this, I think, and do the food industries in the next episode. It's quite long. So we get our waters from natural reservoirs, rivers, lake, but these waters are always subjected to contaminants, either natural contaminants or contaminants that we got through human actions. So when the water is rerouted to our municipal pipelines, these raw waters undergo a couple of processes. Now, I found a good video on water treatment. I'll leave the link in the description so you can study it. But roughly, you've got 4 stages in the process: flocculation, sedimentation, filtration, and disinfection. Now, take note that these don't need to be in that order exactly. It depends on the facility and depends on the design decisions by the engineers. Anyway, flocculation, or alum flocculation, sometimes they're called, it's the first stage. A floc FLOC is a type of colloid suspension. So think of it as jelly, gooey substances that you can find in raw water. So the process of flocculation will agglomerate these flocks or makes them bind together. And to do this, we use a flocculant, which is a polymer that can make the agglomeration or make the flocculation happen, right? Basically, it gets stuff to bind together. A common flocculant is alum or aluminium sulfate. So you put this alum into the flocks and the flocks will agglomerate, then they become denser than water. And when they become denser, they settle down as a sediment. That sedimentation is the next stage. Here we don't just sediment the flocks, but also all large bodies like wood or stones. In the next stage, we have to filter the water. The water by now is relatively clarified. You won't have big chunks of stuff in it, but it's still got microscopic solids and of course microorganisms, which can be harmful. So in the filtration stage, the water is ran through layers of sands and activated carbon. And then the final stage is disinfection. We use chlorine or ozone or UV to get rid of the microorganisms. Well, most of them, not all. You still should boil the water that gets into your home before you drink them. Then on the other side of the municipal use, you have wastewater that needs to be treated. I'll leave you another good video for this one. Study it. Study the relevant textbook sections on this as well. Generally, you have three major treatment stages: separation, biodegradation, and disinfection. The primary treatment is separation. And it's called that because we need to separate larger objects, solid objects, from the water. We do this to protect the machines in the subsequent stages. So you have your screening chamber or bar screen, grid chambers, and all of those for removing floating materials. It's called preliminary treatment or pre-treatment, depending on the articles you study. At the end of it, you have water which contains only microscopic contaminants, theoretically all organic, nothing more than 10 microns. Then the water goes to the secondary treatment, which is biodegradation. The effluent from the primary treatment goes into either a trickling filter or activated sludge systems. Actually, there are more options, but let's focus on those two. In trickling filter system, you have a bed of materials, both natural materials like rocks or gravels and artificial materials as well, like plastics. So we let microorganisms to grow within this bed, right? So you have biofilms throughout the bed. Then the water is trickled through the bed slowly. So during this process, the microorganisms that live in the bed will degrade the biomaterials, essentially cleaning up the water. So that's the trickling filter system. The activated sludge system uses sludge, which is soft, thick, and wet materials, and it has an army of microorganisms growing in there as well. So you activate the sludge in the aeration tank. So you do this by injecting oxygen into that mix. So when the water goes through this aeration tank, the microorganisms will degrade the biological materials. The tertiary treatment is some combinations of filtration and disinfection. So this is the final stage before you can discharge the water into the river. The methods are similar to the earlier potable water treatment. You use chlorine or UV or ozones. And all of this is to make sure that the water is safe to be released into the environment. All right, that's it. So study the relevant sections in the textbook and the videos that I'll give you. While you're studying, take a breath and reflect on what you have just learned here. We sometimes take for granted that we have water when we open the tap, and that we use the toilet, and that the whole sewerage system is working. I know you might get laughed at if you say that you are grateful that your toilet is working, right? I know how it sounds, but those who are laughing at you probably haven't learned what you just learned, right? You're not like them. You are science students, and you study all this. You know things work because of the scientists, because of the engineers, and because of the honest blue-collar workers who operate these treatment plants, right? I mean, I have talked to students who come from countries that are in conflict. And in those countries, they can't access things as simple as treated tap water and wastewater systems. And you know what happens? People die, of infections. Mothers cry because their little babies are dying because of the dirty water. So yeah, we should be grateful because the toilet system works. We should be grateful that our taps have got water coming out. And then you pray for the workers who withstand the smell and the tiredness when running this treatment system, even if they don't get paid much. They are closer to God than the rich industrial managers who contaminated the rivers. All right, okay, that's all. I'll talk to you guys later in the next episode. Barakallahu li wa lakum. Assalamu alaikum warahmatullah.